thanks everybody for joining us. This is MCI's Lemonade Stand series. If you haven't joined us before, it's really just a thing that we started in the early days of COVID to try and bring together the research community in some way and provide resources that are informative to those that are in the male contraceptive space. That can be the advocacy angle, the research angle, or other angles. And today we'll be talking about specifically the angle of trainee support. A little housekeeping before we go. You can check out old Lemonade Stands, our previous ones, on our YouTube page, where we cover some specific things about male contraceptive development. We also talk about MCI programs, as well as other male contraceptives in development, like hormonal methods. To get started, I wanted to talk about the importance of what we'll be talking about today, the trainee support mechanisms that we have in the fellowships. MCI has a variety of research funding mechanisms, but support for trainees, I think, is especially important. And it's important for us because it's important for sustaining the field. I think you're all aware on this call of how small the field of male contraception really is. And so it's important for us to create a human pipeline of researchers, as well as a pipeline of molecules or methods of male contraception, so that we're sustaining the future while at the same time supporting the research that's being done today, oftentimes by the graduate and postdoctoral trainees out there. So the ways that we do trainee support, we have a success program where trainees can apply on our website for a variety of things that they might find they need some kind of financial or other support for. Things like professional development or conferences or courses, things that advance both them and their male contraceptive careers. But the main topic of what we'll talk about today is really our fellowship program. That's really our flagship trainee support mechanism. It's both graduate students as well as postdocs. There's 2 years of support provided, and applicants work with a PI to write and then submit a proposal where they carry out their own individual research. The idea, of course, being that they get experience in writing and executing a proposal, but also in really developing their own research strategy and plan. Our 2019 cohort of fellows has actually just finished up all of their fellowships. You've got them here on the call with you. And here we're gonna have them present their work, and then we'll have a little bit of a group discussion afterwards about the usefulness, the importance of trainee support and what this has meant. So I'll kick it off here in just a second, but I do want to introduce everyone individually before we start. We'll have Melanie Balbach presenting first from Weill Cornell in Manhattan. Then we'll have Jae Yeon Hwang coming from Yale, and then we'll have Max Lyon from WashU in St. Louis. And overall, they'll all present their project, what they submitted to MCI, what they're awarded a fellowship for, and then how things have gone over their 2-year fellowship. So with that, I'll hand it off to our first presenter, Melanie. Please take it away.
So to this audience, I don't really have to explain how important it is to develop new methods of contraception. So I figured I'd just jump right in. My PowerPoint wants to do what I wanted to do. There we go. So when mammalian sperm enter the female genital tract, they are in an immature, non-capacitated state, not able to fertilize the oocyte. They undergo the process called capacitation as soon as they come in contact with bicarb in the fluids and in the ejaculate, that then activates the enzyme our work is working on, which is soluble adenylyl cyclase. And we just abbreviated shortly as SAC, which I'm going to do for the rest of the talk. SAC is then catalyzing the reaction of converting ATP into cyclic AMP, which then initiates the signaling pathway, converting the non-capacitated into the capacitated sperm, being able to fertilize the oocyte. Soluble adenylyl cyclase is not only crucial for capacitation, it also regulates sperm motility. So here on the left, you see a wild-type mouse sperm tethered to the glass surface with its head and the frantically moving flagellar beating pattern in the presence of bicarbonate. In contrast, when you compare the SAC knockout sperm, you see that it's barely moving even in the presence of bicarbonate. It just shows small vibratory movements. And then that defect can be rescued by adding cell-permeable CAMP back to the cells. Since SAC is essential for capacitation and also motility, the male mice are completely infertile. And in 2019, there were also 2 Iranian farmers reported that have a mutation in the SAC locus that have a similar infertility phenotype and also the similar motility defect as the mice. The idea that SAC would be a good contraceptive target is actually not new. They, of course, already discussed it when SAC was initially discovered, but the idea was quickly discarded when people realized that SAC is actually not only expressed in testes, but rather ubiquitously expressed. But the fact that these Iranian farmers are perfectly healthy despite their infertility and a few kidney stones motivated us to rediscover SAC as a contraceptive target. Luckily for us, there was already a small molecular inhibitor available that one of the previous postdocs found in a high throughput screening, which was called LI1, the abbreviation of the person who actually found it. And you see that LI1 dose-dependently blocks in vitro fertilization in mice. We also had a crystal structure available of LRA1 and the SAC protein. So with a local drug development institute, we set out the task to develop LRA1 into a safe, non-hormonal, on-demand male contraceptive pill. To assess the potency of the newly developed inhibitors, we used a hex cell line that overexpresses SAC. So what we do is we treat those cells with different concentration of the SAC inhibitor or DMSO as control, and then we block the phosphodiesterases with IBMX. We then let the CAMP accumulate for 10 minutes and quantify the CAMP via an ELISA. So here on the right, you see the dose response curve of our starting compound, which was LRA1. Then the next the big milestone we made was with the compound 10-229. So they're just named in numerical order. So you just have to deal with those names. I know they're not quite handy. But what is nice to see is that 10-229 compared to LRA1 had an about 1,000-fold increase in potency. And we used 10-229 to show some basic principle that SAC is actually blocked in mouse and also in human sperm, and we recently got that published in Molecular Human Reproduction. I also tested 10-229 in vivo in mice, and I was able to achieve a 40% decrease in fertility. So this was already a very good step into the right direction. So we kept working on increasing the potency of the inhibitors and also the drug-like properties. So one of the current best inhibitors of that last generation we have is called 11861. So you see that the potency is even further improved, and it's also more soluble and also has a higher affinity towards the enzyme with a much lower off rate than the other compounds. So for the rest of the talk, I will focus on 11861. First, I had to confirm that 11861 blocks capacitation in vitro. So here in mouse sperm, you see that during capacitation, there is a 2-fold increase in intracellular CAMP. And that increase in CAMP was fully blocked with 5 nanomolar 11861. Similar in human sperm, you see that 2-fold increase in CAMP, which again was blocked with 5 nanomolar 861. This is quite remarkable considering that human sperm had already come in contact with bicarbonate during ejaculation, and we're still able to block them afterwards. I also did some washout experiments where I diluted the sperm with the inhibitor 100-fold, and you see that the reaction is still blocked, both in mouse and also in human sperm, confirming that the inhibitor has a very slow off rate. 11861 also perfectly blocks pKa activation, tyrosine phosphorylation, acrosome reaction, hyperactivation, and in vitro fertilization. But due to time reasons, I will not show you that data. Instead, I want to quickly focus on the motility analysis I did. So here on the left, again, you see a basal wild-type mouse sperm just tethered with the head. And then here in the middle, you see how the beat frequency increases when the sperm come in contact with bicarbonate. Now, 11861, as you see here, not only blocks that bicarbonate-induced increase in beat frequency, it lowers the beat frequency even further. Similar for human sperm, again, you see the basal beat frequency and the beat frequency activated by bicarbonate. And again, also in human sperm, the increase in beat frequency by bicarbonate is only blocked, it is further reduced. So this all worked perfectly in vitro, so I wanted to test if the inhibitor also works by systemic exposure into the mice. So for that, I developed something I called ex vivo capacitation assay. So for that, I injected the mice with 50 milligram per kilogram 11861. And then after defined time points, I isolated the sperm. Those sperm were then subjected to non-capacitated or capacitating conditions. And then after a distinct time point of 12 minutes, I quantified the AMP concentration. So on this graph, to make it a little bit easier, I normalized the CAMP increase to the respective non-capacitated control. So here, the 0-hour vehicle control, again, you see that twofold increase in CAMP. So essentially, everything that is higher than 1 shows an activation. Then if we look at the different time points, you see that 1, 9, and 24 hours post-injection, the CAMP response is still blocked. And then sometimes between 24 and 48 hours, the response comes back. And then 72 hours later, it's fully recovered. So 861 not only blocks capacitation after systemic exposure, it is also reversible. From those similar conditions, I also recorded videos of the sperm. So here on the left, you see video of sperm that were just injected with vehicle, 1 hour post-injection without bicarbonate. And then you see they got much faster when I incubated them with bicarbonate. Strikingly, 1 hour after injection with 50 milligram per kilogram 11861, the sperm are barely moving. So they're essentially looking like sac knockout sperm. They're just doing that small vibratory movement again. And if one would consider that maybe the sperm of a male taking that inhibitor would look similarly, it's hard to imagine that they would make it to the oocyte or even make it past the cervix. And then again, you see 24 hours post-injection, sperm pretty much recovered and gained their motility back. Of course, what we really want to know is if 11861 is efficacious in mating experiments as well. So for that, I treated the male mice either with vehicle or 50 milligram per kilogram 11861 by IP injection. I waited 1 hour so the inhibitor could reach its target. And then I mated the males with female for the subsequent 11 hours. And then I separated the males and the female again, and then waited 24 days to see how many pups I could. So what you can see here that for the vehicle control from 45 different matings, I reached a pregnancy rate of about 45%, which was reduced to 20% with 50 milligram per kilogram 11861. So you can see here that 11861 blocks fertilization in an acute mating. Of course, what we want to reach is 0% fertilization rate. So by trying to achieve that, I reduced the mating window to 8 hours. So I did the similar approach, vehicle injection or injection with 11861, and then after combining the males and the females after an hour, I separated them again 8 hours later. And then again, I was waiting for the pups. So in this graph, you can see that by doing this 8-hour mating window or 9 hours with inhibitor in total, I was able to reduce the pregnancy rate down to about 7%. So this means that out of the 40 matings I did, I only got 3 females that were pregnant, while 2 of them were even subfertile. So currently, I'm waiting for a new batch of inhibitors so that I can increase the dose to 100 milligram per kilogram to maybe get this down all the way to 0. And then additionally, we are getting a lot of inhibitors with similar characteristics as 11861 that we're also going to test in preparation to start clinical trials. So with that, I want to thank my whole lab, the Drug Development Institute, which were unbelievably helpful in making those great compounds for us. Clemens Deckborn at the Uni Bayreuth is doing the crystal structures for us. And of course, the biggest thank you has to go to Merck Contraceptive Initiative for their support for the last 2 years. It was a lot of fun, and I hope we'll work together in the future.
Thank you, Melanie. That's too kind. I'd like to open it up for a quick, see if the audience has any questions before we move on.
Yes, hi, Melanie. Very nice work, very nicely done. Great set of data there. Just a speculative question for you. In the females who became pregnant despite the presence of 11861, do you have any kind of mechanistic suppositions as to why those mice were because clearly in the ones where they got out. I mean, there was no question they were very pregnant. There was a litter of 9 pups, I think one of the data points was. So that suggests that there was little or no effect on the male side. So do you have any kind of speculation on mechanism for why that might have been? Thank you.
So the difficulty with the mice is that their mating window is really poorly defined. So we actually don't know if they start mating right away after I put them together with the females, or if they may be doing it right before I separate them again in the morning. So it could just be that the ones that got pregnant just had sex really, really late. So the effect of the inhibitor had already worn off. So what we're trying to do is start videotaping them actually to kind of see if maybe that is the case. And we also have now the Christmas mice in the lab, so the ones that have green and red fluorescence in the mice. So with those, we also want to test how far the sperm are actually getting after the sperm got the inhibitor, so we have a better idea where that inhibitor effect is actually coming from.
Great, thank you.
Melanie, this is Greg Kopf. Really nice presentation. This is not a question, but I just want to, ask you just make a comment, because when you're talking about 11861 in one of the experiments you talked about, basically what you were getting at was a slow off rate of the compound. And just like you to talk a little bit, to the group about the significance of a compound like that for the development of a male contraceptive, especially affecting a target like this one.
I mean, that's essentially what you want to a certain degree, right? So you want something that comes off very slowly, but not too slowly, so it can actually have long-term damaging effects. So we're aiming a very delicate balance here. So we want it to last long enough so it actually covers the whole fertilization window, which will be even trickier when we actually get to human because it's way longer. So just reports of even 4 days in total. But then after the inhibitor has done its job, we also want it to come off again and then block the inhibition and also not harm the sperm that are coming after that. So it's kind of, I think we all underestimated in the beginning how difficult that drug development process actually would be. And this is how TDI is so incredibly helpful, because they know what they're doing, and you essentially need people like that on board to be able to successful, because in the end, what it comes down to is a good compound, really, that works as perfect as you want it to be.
Yeah, thank you. I think it's such a critical component for identifying potential drugs that could function in this manner. Thanks for that.
Yeah, absolutely.
We're going to go ahead and move on to our second speaker. If we have some time at the very end of everything, we can move back around and bring some more questions around. But I want to move on to Jae Yeon Hwang, coming out of Yale, who's going to talk about his work with Casper. Jae Yeon, feel free to take it away.
Okay, thank you. Just 1 second. So can you see my screen now?
Yep, you're good to go.
Okay. So hello and good afternoon. I'm Jae Yeon Hwang from Jinjong's lab in School of Medicine. And today I'd like to talk about my fellowship project regarding developing a screening platform of Casper Epsilon for the male contraceptive development. Oh, just 1 second. So actually, as Melanie explained, the ejaculated mammalian sperm cells getting fertilizing ability gradually in a female reproductive tract, called in a process called capacitation. And one of the specific phenotype observed from the capacitated sperm is they change their motility pattern called hyperactivated motility. This motility pattern is characterized by the asymmetry flagellar beating with increased amplitude. And for the last 20 years study, this hyperactivated motility is achieved by the influx of the calcium ion through the sperm-specific calcium channel Casper. So previous studies clearly demonstrate that this hyperactivated motility is essential for male fertility, and the absence or defects on the Casper channel cause the impaired hyperactivated motility, which finally caused the male infertility. So from these research and previous studies, I thought if we target the Casper channel and compromise the calcium influx in sperm cells, that will be a good candidate for the male contraceptive system. Indeed, Casper channel is quite a complex ion channel component because this protein is composed of multiple proteins and currently over 10 subunits have been reported. And as you can see from this diagram, the 4 accelerated transmembrane subunit Casper beta, gamma, delta, epsilon has very large extracellular domain. And recently our group reported that this extracellular domain form very unique pattern and unique structure, looks like a canopy at the extracellular region. And previous study reported in 2019, there is one report regarding the mutations on in-frame mutations on the Casper epsilon caused the male infertility from the CRISPR group. So from these observation and studies in this fellowship program, I suggest the study if we target the Casper epsilon, then it can be used for the male contraceptive systems, and to develop, I plan to develop the screening platform for the Casper epsilon contraceptive systems. So in this program, I aimed at 2 parts. The first aim was generate the Casper epsilon knockout model and characterize the phenotype to confirm whether this is really important for the male fertility. And the second aim was the molecularly determine the function of the extracellular domain of the Casper epsilon in sperm cells. So by using the CRISPR-Cas9 genome editing system, we generated Casper epsilon knockout model, and we found these the knockout males are 100% infertile, and we found their motility pattern is severely damaged because they fail to develop hyperactivated motility compared to the wild type sperm cells after using 90 minutes capacitation. So because the hyperactivated motility is highly associated with the calcium influx, we measure the calcium influx in the Casper epsilon knockout model, knockout sperm cells, and we found Casper epsilon knockout sperm cells have severely impaired calcium influx, and this is caused by the absence of entire Casper channel components. So from this result, we can conclude that the Caspar epsilon is the proper target for the Caspar for the male contraceptive. In a second aim, actually this part is the contents what I spent majority of time during the last 2 years. So I want to identify and understand what's the molecular function of the extracellular domain of the Caspar epsilon, because currently it is not possible to reconstruct the Caspar channel in the in vitro system. So instead of generate the Caspar channel in heterologous system, I focused on the purified proteins of the extracellular domain of the Caspar epsilon. Indeed, Caspar epsilon is around 950 amino acid, but their overall 900 amino acid is occupy the extracellular domain of the protein. And recently published paper demonstrated that this large extracellular domain is composed of 5 groups. So actually for around 6 months, I spent my effort to purify the full length of the extracellular domain with keep changing the tag or secretion system or other expression system, but unfortunately, all of them were not successful. So alternatively, I tried to purify the truncated proteins of the extracellular domain of Caspar epsilon. And as you can see from here, a full length and like a 660 amino acid extracellular domain, or 440 amino acid, or around 200 amino acid. I test from the testing of the solubility of the extracellular domain of the Caspar epsilon. I found these 2 truncated Caspar epsilon extracellular domain proteins are a little bit better to purify the proteins, because this S means the solubilized fraction and this P means the pellet fraction. So in other words, insoluble fraction. So what you can only purify is from this only S fractions. So compared to the other 2 recombinant proteins, only 440 amino acid or 215 amino acid proteins were doable to purify. And actually, even though I a little bit increased the scale and testing the perform the purification, but still the purity of the protein is not that much good. But still, the purified fraction still contains some target proteins which can be detected from the chromatin blue gel staining level. So because previous studies revealed that this extracellular domain contains a lot of cysteine, and one study suggests the cysteine might serve as may give some function as an isomerase, which means that making some disulfide bond on the protein. So to test the possible isomerase activity of this purified extracellular domain of the Caspar epsilon, I designed kinds of some interesting experiment, because if they have I hypothesize that if they have a real isomerase activity, then they can interact to the reduced sperm proteins. So I pre-incubated with a sperm with a BSA to induce some efflux of the cholesterol, and then induce reducing by using the T-CEP. And after 30 minutes of the reducing, I washed out the any reducing reagent and incubated with these recombinant proteins. And then I checked whether these recombinant protein can be interact to the sperm cells. And based on the immunoblotting result, I found this recombinant protein is reduced, successfully interact to the sperm cells after reducing. That means this recombinant protein could have some isomerase activity to interact with the sperm proteins. And from this result, now I think actually this recombinant protein itself can be used for the screening platform to prevent or enhance the disulfide bond formation together with the other Caspar auxiliary subunits. So this is pretty much what I did for the fellowship program. And last but not least, I want to express my huge appreciation to all of my lab members. My super PI, Jinju, always gave me a great suggestion and give a good direction. And actually, the Hwang and Jongnam Gu worked with me for the initial mouse characterization, and the Luke supported the protein purification process. Definitely, it was my great pleasure to get supported from MCI for the last 2 years, because with the support from the MCI, I was able to publish 3 papers for 2 years and complete 2 other projects. But although this achievement is really important for me for the future career, but I feel MCI gave me a great opportunity to think deeply how the basic science contributes to the society. This enabled me to expand my research interest. So again, I deeply appreciate MCI Foundation. Thank you.
Thank you. Appreciate that. We'll open the floor for some questions if anybody has any.
Yeah, I have a question. This is great, Cop. Very nice presentation. So the recent work by Lin, which you referenced in Nature, their cryo-EM work, positions Caspar Epsilon, with the other subunits. Is this now, these data that are out there, does this give you, a greater opportunity to develop a series of inhibitors that might not only affect epsilon, but might also affect epsilon's interaction with some of the other subunits of Caspar?
Yeah, definitely, I agree. And indeed, I didn't include the result in here, but I also tried the other extracellular subunit, Caspar Delta. But the reason I didn't include the result in here was because the solubility of the protein was quite bad, much worse than Caspar Epsilon. So, but as you asked, I think the other components could be also good candidate, because all other 4 oxygen subunits have over 600 or 700 amino acid of extracellular domain. So they could be very important and good candidate for the male contraceptive development, I think.
Thank you.
I have a question related to those extracellular domains. You mentioned that there's solubility issues with delta, and then I see on this slide here for epsilon, there's some solubility issues when you get into the longer pieces of the extracellular domain. Do you think that the domains are important for their own stability to interact as a complex, or what do you think is the source of that instability, being as they're all extracellular and you would think they would be sort of soluble?
Actually, this is my personal experience from the experiment with the Caspar Epsilon or Delta. But I keep continuously found was when I overexpress the full length of the extracellular domain, then even though I denature the proteins, there's some aggregates on the top region. That means if I overexpress the full length of the, or relatively longer size of the extracellular domain, because they harbor much more cysteine residues, and they form very, very strong disulfide. This is my guess. So that prevent kinds of some normal structure formation and solubilization of the proteins. So that would be one reason, because they have a lot of cysteine residues and make some unrelated disulfide bond with the other protein itself. So that would prevent, I think that would prevent the solubility of the proteins. And well, because in here I tested like a C-terminus based purification, and recently I also tried the N-terminus purification. And maybe depending on the domain reason, I think the solubility could be changing. But currently, the beta-propionyl reason would be the most difficult reason to be purified. This is my experience from the.
Thank you, I appreciate that. All right, we're going to go ahead and move on to our final speaker, Max Lyon, coming from WashU in St. Louis. Max, feel free to take it away.
Okay, showing up all right?
Yep, you're good to go.
Okay, so thank you all for coming. I'm going to be talking about my fellowship research, which was on looking at SLO3 as a non-hormonal target for contraception. So as the last two speakers covered, sperm have to undergo capacitation in order to fertilize. In the epididymis, they're stored in an inmotile and infertile state. When they enter the female genital tract and interact with the seminal plasma, they become motile, but they're still infertile. In order to become fertile, they have to undergo capacitation, which is a series of processes, including several steps, one of which is a hyperpolarization of the membrane to a more negative potential. There's also an influx of calcium ions, and then two processes, hyperactivation of motility and an exocytotic process called the agrosome reaction, both of which are needed for the sperm to actually fertilize. Only once all these processes occur are the sperm actually fertile. My project is looking at the factors that regulate hyperpolarization and then seeing if we can block that hyperpolarization to prevent fertility in human sperm. To go over what's known about hyperpolarization, I'm first going to start with the mouse model because that's been very well characterized. And we know that in mice, hyperpolarization is dependent on a potassium channel called SLO3. This channel is expressed only in sperm and allows potassium ions to flow out of the cell. And it is also activated by increasing intracellular pH. So if we record the potassium currents in human sperm, we find that they're voltage activated and they're stimulated by pH increases by things such as adding extracellular ammonium chloride. And if we knock out the SLO3 channel in mice, we lose that potassium current as well as the pH activation. When we incubate mouse sperm in capacitating conditions, they go from a resting membrane potential of around -40 millivolts to a final potential that's hyperpolarized to around -65 millivolts. And this is caused by the SLO3 channel opening and allowing potassium ions to flow out. This loss of positive charges causes the inside of the cell to become increasingly negative. If we knock out the SLO3 channel, we lose this hyperpolarization during capacitation, and the mice lose the ability to undergo the agrosome reaction. They have some slight defects in motility, and ultimately they're infertile. Now, this indicated to us that SLO3 is a valid target for contraceptive research. However, there are some questions on the specifics of how this process occurs in human sperm. And one of the main reasons for this is that when the potassium currents in human sperm were first examined, they were found to be activated by increasing intracellular calcium. And this has traditionally been a hallmark of the related SLO1 channels and not of SLO3 channels. However, when the human SLO3 channel was expressed heterologously, it was found to also be activated by intracellular calcium, unlike the mouse SLO3 channel. And this could indicate that either the channel has evolved separate regulation or that there might be something else present in human sperm. And so the question of what role is SLO3 filling in human sperm has yet to be answered. The goals for my project were to verify that SLO3 regulates human sperm membrane potential. One of the blocks that's existed in this for this research is that there are no published selective inhibitors for human SLO3, so it's been hard to distinguish the contribution of SLO3 and SLO1. We also want to verify the role of SLO3 in human fertility. Unlike things like CATSPR, there are no known natural loss of function mutants for human SLO3, so we haven't found a patient where we can say they're infertile because they have no functional SLO3. And then finally, we want to confirm that human SLO3 is a viable target for contraception. And because we can't generate any human mutants, we instead go for the inhibitor approach to try to find something that can block SLO3. And to do this, we partnered with the lab at Vanderbilt University, and there they developed a high throughput screen using HEC293 cell line that was stably expressing SLO3, along with an auxiliary subunit gamma-2. This high throughput screen used a Thallium flex assay, and the way this assay works is you take your cell line that is expressing your channel of interest, in this case SLO3, you load those cells with the dithallose, and then you perfuse a buffer containing thallium ions over those cells. Thallium is a monovalent cation that flows through potassium channels such as SLO3, and when thallose binds to it, that causes an increase in fluorescence. You're able to then add small molecules to this and see a decrease in that fluorescence to show you that you're inhibiting the SLO3 channel. They tested over 50,000 compounds using this high throughput screen, and from this they found 5, which they then handed to us to test with our other methods. And for this presentation, I'm only going to be talking about one, which is inhibitor 110. So the first thing that we did is we wanted to validate that we were inhibiting the SLO3 channel using patch clamp electrophysiology, which is a gold standard for these types of experiments. To do this, we took that same HET293 cell line and then attached an electrode to it and exposed it to a current, to a voltage, which in this case was 100 millivolts, and recorded the resulting currents over an increasing range of inhibitor concentrations. From this, we saw that we did inhibit the SLO3 channel in a dose-dependent manner. We also tested the effect of inhibitor 110 on that related channel SLO1 and saw no effect even at high concentrations. We also wanted to see if the target of the inhibitor 110 was the SLO3 channel or that gamma-2 subunit. And to test this, we co-expressed SLO1 with gamma-2 or with a related subunit beta-4. Both gamma-2 and beta-4 are found in sperm and interact with both SLO1 and SLO3 channels. From the dose response curves that we built in these experiments, we calculated the IC50 of SLO3 as 1.3 micromolar. This is the concentration where we inhibit half of the current going through those cells. And this is very high compared to other SLO3 inhibitors that are currently used, which are typically used at about the 100 micromolar range. We also found that that inhibitor 110 was at least 45-fold selective for SLO3 over SLO1. The next step was to then test this inhibitor in human sperm to see if we were blocking the potassium current. So we did, two members of my lab, Ping Li and Juan Freya, did whole cell patch clamp on human sperm. And they saw that the potassium current in human sperm was calcium activated and was almost entirely blocked by inhibitor 110. Furthermore, this block did not seem to be calcium dependent, as at every concentration that we tested, the potassium current was almost entirely abolished. We also wanted to check if the inhibition was pH dependent, and this is important because as sperm move through the female reproductive tract, they're exposed to a very large pH gradient as they progress further in. And we saw that there was no difference in inhibition at pH 7.2 or at pH 8, indicating the block was not pH sensitive. And this is also very important for our next set of experiments, which was looking at the effect on membrane potential. The way that we measure membrane potential is using spectrophotometry and a fluorescent dye called DISC35. DISC35 is a positively charged dye, which is cell permeable. What we do is we load the sperm with this dye, which moves into the cells because they're negatively charged. We record our baseline fluorescence of the solution outside and the amount of DISC that is there. We then add the potassium ionophore valinomycin. And as the potassium flows out of the cell, the DISC35 goes in. As we approach the equilibrium potential of potassium, which we calculate based on the Nernst equation, and the concentration of potassium inside and outside of the sperm. We then add increasing steps of potassium chloride, driving more of that DISC35 out. And from this internal calibration curve, we back calculate our initial membrane potential of these cells. We first confirmed the same thing which has been published many times before, which is that incubating sperm at a high pH causes a hyperpolarization, in this case from around minus 38 millivolts to around minus 43 or 45. And when we added inhibitor 110 during this process, we saw that we eliminated that hyperpolarization of human sperm. And then once again, to confirm that it is SLO3 and not SLO1 that's important for this process, we also tested 3 SLO1 inhibitors, Paxilene, Ibiriotoxin, and TEA, and found that none of those 3 inhibitors were able to prevent this hyperpolarization. The next step was to look at the effects on the hyperactivated motility in the agrosome reaction. We found that at concentrations as low as 0.5 micromolar, we were able to significantly inhibit hyperactivated motility in capacitated human sperm. To test the agrosome reaction, we induced it with a calcium ionophore A23187 after overnight incubation in capacitating conditions. And we saw that we were able to significantly inhibit the agrosome reaction when inhibitor 110 was added during the induction step or during the overnight incubation with incapacitating conditions. In conclusion, we have characterized the first selective and potent inhibitor for human SLO3. We have confirmed that human SLO3 carries the potassium current in human sperm and regulates hyperpolarization. We confirmed that human SLO3 is involved in hyperactivation in the agrosome reaction. And together with some recently published data we have that showed that hyperpolarization of human sperm correlates with success in IVF, we have determined that SLO3 is a valid target for contraceptive development. And furthermore, we have the tools to start building these contraceptives. And this moves us to my future directions for this project, which are to test the effect of second-generation inhibitors on capacitation. We currently have over 200 derivatives of the first-generation compounds that we tested, all of which are designed to be more potent and more selective for the SLO3 channel. We also want to see if we can use these inhibitors to block fertilization in mouse using IVF and intrauterine insemination. And at some point, we would also like to test penetration assays using humanized eggs. And this is important because the mouse and the human model are pretty different when it comes to the ion channels and how they're regulated. So we want to mimic the human model as close as we possibly can. And with that, I would like to thank everyone in the Santee Lab who's helped me out with this project greatly, as well as our collaborators at the Vanderbilt University and MCI for your support for these 2 years. It's been great having all of your resources available.
Great, thanks, Max. I'll open the floor for any questions.
Yeah, hey, Max, this is Ted Benson. Really nice data set there. Thank you, great presentation. I'm curious about the 110 compound and its various derivatives. You probably don't have this data for the derivatives yet, but you might have it from the 110 because it came from a library. Are there other channels that it's known to affect other than slow? I mean, do you have any information about that? If there's any sort of related channels that it interacts with, or is it highly specific to start with? I mean, you've gone more specific with it, I get that, but I'm just curious.
Yeah, so the main channels that we were testing for the first round was SLO1 because of its proposed role in human sperm. Another channel that we tested was against HERG, which is a cardiac potassium, or which is another cardiac channel. It's a very common off-target, and that was one that we were most concerned about. And all of the first generation compounds that we tested also substantially blocked HERG. Okay. So the main goal with the second generation compounds that we've developed so far was selectivity over HERG. And of the ones that we've found, those 200 compounds, I think most, if not all of them, were at least 1,000-fold selective over HERG. So that was the first milestone that we had. And so that's the main thing that we've been testing against. And then we just recently started testing those against SLO2, which is, there's 2 variants of that, SLIC and SLAC, which are also important cardiac channels and are also related. They're the other members of the SLO family. And we've found pretty good selectivity over those so far as well.
Good, great. I'm glad you're getting away from HERG. That's encouraging.
Yeah, that was a major milestone for us.
Yes, and a very important one. Thanks.
So Max,
so what happens when you inject this stuff into mice?
Yeah, so that's another question that we had. And we have done a little bit of preliminary experiments in mice where we've done intrauterine insemination. And we just did that overnight with one of our first generation inhibitors. We didn't notice any deleterious effects. We had to sack the mice the next morning though. So we weren't able to see if there were any long-term consequences. We are definitely planning on doing more experiments in mice with the second generation compounds once we finish getting them characterized though.
So Max, this is Greg Kopp. Very nice presentation. You made a comment that you weren't able to find any human genetic mutations that would result in infertility. How did you go about looking for that? What databases did you look at?
So our lab published a paper, and I believe it was 2018, looking at different allele variants of human SLO3. They found one variant which was not very uncommon, that had increased calcium and pH sensitivity. However, we haven't found a patient that actually was infertile and was tested and found to have SLO3 deficiency. I haven't actually.
Yeah, no, go ahead. Oh,
sorry, yeah, I haven't looked through those databases to see if there are any major deletions or any ones that we would assume would be loss of function. But one of the problems is when we're doing fertility tests, we only look at sperm precapacitation. And the effects that we see of SLO3 loss, we don't think would affect those parameters. And so it can be a problem where patients are presenting as normospermic, but they actually have a SLO3 defect that prevents capacitation.
Right.
And that's been another drive of our lab, is to try to get fertility clinics to actually look at those parameters and look at hyperpolarization as an indicator of sperm fertility.
Yeah, no, I was going to say that, there's, if you hadn't looked, there's a faculty member up at Mass General. Her name is Stephanie Seminara. I don't know whether you know the name. She's professor of medicine at Mass General, and she's reproductive endocrine. And she's got access, not only her, but she has networks with other researchers that have very large databases, infertility databases and patients and things like that from all over the world, if I remember correctly. Now, I don't know how much data she has on the male side, but it's somebody might want to reach out to.
Thank you. Yeah, that's great. I will definitely try reaching out.
Sure.
Greg, thanks for telling us that.
Yeah,
This is Celia. So yeah, we are like, regarding that, we are also looking at normal spermic infertile patients, and we are doing exome sequencing from those patients to see if we can find, especially the ones that fail to hyperpolarize, because we have that data. So, but it would be great to contact these women here, so that will give us like more data.
Yeah, I mean, she, I don't know how much information she has immediately, but she could connect you with others potentially. I know that there's some data, some scientists at University of Utah that have some pretty significant databases as well. I just don't remember the names of the folks there.
Okay,
So,
thank you.
sure.
Yeah.
Awesome. Well, thanks, Max. Thanks for those great discussion points, and thanks for bringing that up, Greg. I wanted to take one moment to do a little bit of housekeeping before we kind of open things back up again, to just illustrate a couple of MCI funding efforts that are coming down the pipeline, specifically for trainee support. So the cohort that you just heard from is our 2019 cohort, and we just recently made our 2021 awards. Our 2022 cycle will be opening in March, April of next year. And we encourage anybody who is a graduate or postdoctoral trainee to take a look. If your research applies, we'd be very interested to have application from you. And I'm also interested to just speak with you beforehand. Happy to talk about MCI's priorities and what we've done in the past with these fellowships. I also wanted to mention we have a annual travel award for an international student. Things have been kind of wonky, as I'm sure everybody gets, for international travel in the last year or so, but the intent of that award is to facilitate collaborations across borders, to allow people to come to neighboring labs, learn new tools and techniques, bring them back to their original lab, or maybe travel to an internationally recognized conference. That's our Beverly Tucker Award. You can find information about that on our website. malecontraceptive.org/funding is where you'll find that. And I also wanted to call attention to the Trainee Success Program that I mentioned. That's support for professional development, courses, also travel grants for conferences. It's just not restricted to international travel. And just to say that that's really a program that's designed to be open to whatever you bring to it. We've supported a variety of efforts for graduate students and postdoctoral fellows there. And it's all with the intent of allowing them to get the skills they need to transition to the next phase of their career. So if you have something that's applicable, you think it might be applicable, you want it to be applicable, please go to our website, take a look at the form, and then reach out to us if you have any questions. We're more than happy to assist you. With that, we've got about 5 more minutes. I'll open the floor if anybody has any questions for any of our panelists. But if you don't, I have some questions. I see Lonnie raising his hand. Go ahead.
Hi, Lonnie Levin, New York. Max, nice progress, by the way. Great progress. Since I guess it was a long time ago when I saw this, but great progress.
Thank you.
My question is, if you're giving this intrauterine, and you may have a goal ultimately to go into the male, but if you give this intrauterine, is there really a drive? Do you have the drive to be slow 3 selective relative to slow 1? If you can make a broad slow inhibitor, you might actually have 2 bangs for your buck and be better at stopping sperm in their tracks. Of course, you want to be away from anything that might hurt the female reproductive tract, but your goal to be slow 3 selective, maybe not necessary, a high bar to reach that you reached it, but it may be a high bar to reach that you may not need if you can target the female milieu.
Yeah, so the main downside of targeting slow 1 is that it's also involved in smooth muscle activity. And so there's a lot of potential downsides if we target that one. And slow 3 is only expressed in testes. So we can be selective for that. And even if we have to use it in the female, there is no SLO3 expression or functional SLO3 channels in the female body. So that's another positive benefit, is that if you can get it onto the sperm somehow, whether it's in the male or in the female, ideally it wouldn't have off targets anywhere else. So that's been one of our big drives about getting it selective, other than just answering the the contribution of SLO3 in sperm. Yeah, thank you though.
Margaret, did I see a hand from you as well? Nope, you're on mute.
Waiting for all the scientists to have their say. This is coming from a different direction. I noticed, though, that several of you advertised the fact that it was a non-hormonal approach that you were pursuing. And I want to know whether you think that that's more certain, of course, to explore, or easier to operationalize in drug or procedure development, or whether it's really a question of greater acceptability, public acceptability. There's been so much negative press about hormonal stuff in female as well as any other context, contraception. So that's just an out of the park kind of question, but I'd be interested to hear why you emphasize that feature of what you're doing, a non-hormonal approach to, and if you see that that's really the way of the future in male contraception.
Well, I can speak to that. I know both Greg and I have been on NIH panels maybe 15, 20 years ago where they started talking about non-hormonal.
You're aging us.
What?
You're aging us there, Steve.
It's been a while, yeah. The issue is the fact that hormones have so many somatic effects in the body. And the idea is to develop the silver bullet that hits the sperm or the egg specifically. You might have noticed that the Gates Foundation has an RFA for grants for non-hormonal female contraception as well. They're very, very specific about that. The idea being that you want to get a compound that's absolutely razor focused on specific cell biological processes required for sperm or egg to develop competence for fertilization without any peripheral effects on other things. You might have noticed from Max's presentation, very focused on making sure there aren't any cardiac effects with the drugs that they have, because the object is contraception, not killing the patient.
Yeah, let me add to what Steve said. I think really it was a really good question, actually. I think from an industry standpoint in this field, as you mentioned, Margaret, that the side effect profile of hormonal contraception for women has always been an issue, continues to be, and it leads to discontinuation. It also has led over the years to liability issues for the companies making these products. And so, moving forward, ultimately, if we're going to get a male contraceptive into the market, even though right now there are trials of hormonal male contraceptives ongoing right now, but they do have side effects. As Steve said, looking for that silver bullet where you take advantage of a unique gene target that's expressed, in the reproductive tract that controls some aspect of reproduction to be the target for contraception. Certainly companies that would look to manufacture and commercialize such a product, my feeling is they're not going to take on ultimately more liability, on the hormonal side. And as Steve said, hormones have a whole host of somatic effects.
Right.
So it's a good question. It's a good question.
There is the deeper voice in the superior chest, but we know about that study.
Yeah.
I want to make sure to give some space for our fellows to chime in if they have anything they wanted to add here.
Yeah, something to just tack on to I think one of the reasons that there's a lot of interest in things like ion channels in human sperm is that there are a lot that are testis specific. So reproductive genes evolve much more rapidly than somatic ones. One example is SLO3 compared to SLO1. So SLO1 is that cardiac channel that I mentioned, and it has about 99 plus sequence homology between different species. SLO3, which has been proposed to be a gene duplication of SLO1, which is only found in the testes, is only about 60% similar. And there's a lot of different ion channels. There's Caspr, there's SLO1, there's I think it's HB1. There's different ion channels that are only expressed in sperm, and so I think that makes them very, very promising targets for contraceptive development, because ideally, if you can hit just them, you'll have very few off-target effects.
May I add something on that? So also, the ion channels by itself are highly druggable targets, and they are located in the cell membrane, and 20% of the drugs actually approved for human diseases are ion channels. So they are likely, be targets for these drugs. So, and if they are sperm specific, is even better.
So I want to chime in also with one little thing about MCIs. We very specifically support non-hormonal methods, and of course, that's where the majority of our funding goes, is directly into the research of non-hormonal methods. And within that, I wanted to say that we see an opportunity in non-hormonal methods because there are such a variety of targets available to create a male contraceptive, and they have a variety of characteristics that can create products with diverse attributes, like something that's on demand. Maybe it's delivered hours before sex and results in a contraceptive effect, or something that is more long-term. Whereas with the hormonal approach, there is exactly one mechanism, exactly one output. And those products can be packaged and delivered in different ways, but fundamentally, there's still the same mechanism occurring. And we think that within non-hormonal methods, there's not only the promise for different packaging, different delivery devices, but also different target selection that can result in products that meet the needs of more users. With that, I see that we're 3 minutes past the hour, so I'm going to go ahead and wrap us up. If anybody has any questions, feel free to direct them to me. I'm just logan@malecontraceptive.org. I'm more than happy to chat with anybody, and I'm sure our fellows will be more than happy to connect with anyone and answer questions you may have. Thanks very much for coming. You can find the recording for this on our YouTube channel later. And please come to our next one. Keep apprised of our updates on our newsletter and social media. We'll see you around. Thank you.